Hello everyone. Glad to see you all here on our 40th anniversary at Stanford. As you can see from this slide, this is we entitled it Past, Present, and Future of FACS. And it is in '99, the 40th anniversary of Lee and Len having been at Stanford. We came here in '59 when the medical school moved down to Stanford from San Francisco. Some of you I know are from those times, and some are from much more recent times, and even up to the present. This is going to be a little slideshow on FACS and some other matters. Now this is a block diagram of a modern FACS with a couple of lasers, actually 3 lasers, I believe, and the droplets are being sorted by the deflection plates, and you'll see the reason for the 2 lasers lines being shown differently later on in the slideshow. This is a standard block diagram showing the piezoelectric crystal, the nozzle, the fluid coming out with the cells in the middle of the stream, obviously, being hit by a laser, and the droplets being formed and being deflected by deflecting plates and collected in tubes below. The stream is beautifully shown here with strobe light. Beautiful. I love these old pictures which date from 30 years ago or so, back in the early late 60s. The stream is being hit by the laser, and it's pulsating due to the piezoelectric crystal shaking the stream up and down, and droplets are being formed down below. In this next slide, you see a close-up picture of the nozzle, one of the old nozzles. These don't exist on anymore. They were hand drawn, probably by Dick Stovall, or maybe in those days by Russ Hewlett. I'm not really sure. Now we see a little bit lower down, we see the stream pulsating very well, droplets being formed. And this is a stroboscopic picture, meaning that the light is flashing in synchrony with the formation of the droplets, the same frequency as the piezoelectric crystal is shaking the nozzle. And so you're seeing thousands of droplets superimposed upon one another in a time picture, sort of. And so you see the absolutely constant formation of thousands of droplets with a particular shape and particular position. In this next slide, we'll see a little bit further below the droplets breaking off, and you see some droplets in between the main droplets, which is a problem that's been avoided in recent years. And of course, we now sort 1 or 2 droplets per sorting pulse. Another picture, I love these pictures, that's why we're showing so many of them, that the droplets are superimposed one upon another, as I said earlier. We'll go to the next slide. And here you see the droplets have been charged at the moment that they're being formed, at the moment of formation. There are 3 or 4 droplets. That's also, I think, not happening in modern times, where we're getting 1 or 2 droplets sorted. Of course, Dave Parks will know that much better than I do, so you can ask him a question about that when you see him, or you can ask one of the other engineers, or maybe probably Mario Roeder will know about it. So here's a customer coming to have his or her spleen cells sorted. This was a picture we took when there was a strike. Maybe Michael Julius will remember that we had to take care of the animals ourselves. There were no animal caretakers working at that time, and so we brought a mouse down and told him what he was going to face. We're going to have another picture of the mouse, and there he is. And he's adjusting the fine focus, be sure that the operator doesn't have to do it, right? So he's going to donate a spleen, probably, maybe lymph nodes, maybe thymus, who knows? But I don't recall. Maybe somebody does. They can tell me about it. I took a slight break there for a pause, and now I'm resuming narration. We see now the first FACS that got made by BD after we had built a kind of a prototype here at Stanford, and gave the license for a FACS to be made for our lab and for John Wunderlich's lab at the Cancer Institute, with a contract from the Cancer Institute at that time. I guess it was Bill Terry who came and asked me if I would be on a committee would I apply for one of these contracts? And we agreed that I would, and therefore I wouldn't serve on the committee because I wanted the committee to be able to evaluate our contract application. And so from that contract came all the FACS in the world. I think they're now, well, not all, sorry, all the FACSes that are produced by BD, which I know the total is something like 30,000, and I don't know what fraction of those is BD, but certainly the majority, probably the vast majority. You see a beautiful picture which was made, I think David Parks was involved in making this picture, but I'm not sure. It may have been Dick Stovall, it may have been Dick Sweet, who was obviously one of the early, the inventor of the FACS as far as the engineering went, because I was involved in telling them what we wanted to have done, and he was able to really make it go. Bill Bonner had earlier made the first sorter, which was based upon fluorescence. So individual cells could be sorted by in individual droplets, and this is a slide of Dick Stovall having deposited individual beads, I think, rather than cells. And this cloner, as we called it, which could clone individual cells by FACS, was developed by Dave Parks. And there's a picture to show that really is an old picture of both of us, Lee and Len. And you still recognize us? We look a lot different now, at least I do. Lee's got her favorite instrument in her hand. She's not about to throw it to you, but I think she's holding it up while the picture's taken before she resumes talking to somebody. This is her an important instrument. And I am showing that I can actually work the FACS years ago. Can't do it now, and some people wonder whether I could do it then. I think I could, but we'll leave that for discussion. I'm going to go to the next slide. There's a picture of the bench, the sorting tube, the collecting tubes. The nozzle doesn't show very well, but anyway, it's a picture of the FACS 1. So FACS, of course, stands for fluorescence activated cell sorting. Everybody here knows it, but this is going to be on the website, and so that's what it stands for. And the early history is spelled out here, that the first successful sorting of cells was in 1968. It was published in '69 in Science. And I think it was Bonner was first author, Barrett, I think, Hewlett, and Herzenberg. Barrett was a technician in those days. I don't think she's here this evening, or this afternoon, or today, or during this meeting. Anyway, then the patent was made and licensed to BD with that NCI, Cancer Institute contract that I'd gotten. The first commercial instruments came out in the very early 70s, 73, I believe. And in the early 80s, the first real clinical application for FACS came about, which was counting number of CD4 cells for AIDS. And this continues to be a very important clinical application, because there are many basic applications that we'll hear somewhat about today, and you probably know about many of them. Each of you knows of the totality of them, and many of you have had a contribution to making these applications work. You can see, I guess, stem cell sorting for bone marrow transplants came along in the early 90s, and I think that's a very important application as well. Okay, so there were other great improvements, not all engineering ones. I wouldn't call this engineering, but of course production of hybridomas was a very important development for the FACS. I used to give talks entitled FACS and Cell Biology Complementary Tool, or sorry, FACS and Hybridomas, Complementary Tools in Cell Biology, or Complementary Tools in Immunology, or whatever. So clearly hybridomas were very important, and here's a slide that was made for me by an artist that Lee met at the University of Wisconsin, and he put the slide together because I was going to China to tell them about making hybridomas. It was in the early 80s, or was it 70s? Gosh. Okay, you can see what it is. I think it was the early 80s. Yeah, because that would have been 20 years ago. That's right, from now. And so here's Mickey and Minnie taking out his spleen, holding it up, and going to fuse it with the myeloma cells using PEG. You get a gush sound, and I remember the Chinese interpreter who said, "Gush, what's gush mean?" And so I had to sort of tell him. And the people who were there, soon after the Cultural Revolution was over in Shanghai, and the Gang of Four were gone, the audience were very interested in hearing what was new in the West, including hybridomas. And the hat medium sort of escaped them a bit, but I think they got the picture. And out of the hat came the hybridomas, and we could select colonies, and we learned later on how to select them with the FACS as well. But the production of monoclonal antibodies really made the FACS work extremely well. And there are now many monoclonal antibodies, CDs, and I think the number of CDs is well over 200 now. Yeah, another very important biological innovation was the isolation and making phycobilly proteins from seaweeds. So here's a picture of some seaweeds that we collected. Probably Vernoy was involved in this, almost certainly. Randy Hardy was. We used to make our own phycobilly proteins from the seaweed collected near Santa Cruz, or maybe nearer to Monterey Bay, maybe in Monterey Bay, nearer to Monterey. I can't recall at this time. I went on some of the trips, but we went so many times I can't remember which one this one came from. But this is invisible light, and the next slide is going to be with UV. It's going to be startling. Here it is. With UV, you see beautiful fluorescence of these antenna proteins, phycobilly proteins, which collect the light with many fluorophores and transfer them by energy transfer to chlorophyll, from which the plants, of course, get their energy, their sugar, and everything else. And we extract these seaweeds, and we get these phycobilly proteins, which can be sorted by not by the FACS, but rather by chromatography. And there's some pictures of that. And we actually isolated them ourselves, as I said, and conjugated them with antibodies or whatever else. And we still do that. we try to get bulk amounts of antibody, make our own conjugations. Another very important innovation, which we use a lot now, is green fluorescent protein, which is isolated from a polyp, not a polyp, so the GFP is actually isolated from a jellyfish, and I have a picture of it. I think the next slide's going to show that. But before I show that, the very interesting, we wanted to make GFPs that would be easily detected in FACS. And Mike Anderson made some mutants of GFP, which we called VEX and VEX, and the mutations are shown here. And you see that the excitation peak of wild type shows 2 peaks, and we can eliminate one with VEX and one with VEX. And the violet excited, or blue excited, of course, is what VEX and VEX stand for. And we'll show a FACS. We'll repeat that earlier diagram we had in a moment to show how we detect them separately. Slide to come up. And there's the diagram. We haven't got the jellyfish yet, but we'll find it later on. So you see that here we make use of the krypton violet excited line, 406 nanometers, and the argon laser putting out a 488 line. And so that separately excites the GFP into 2 different wavelengths. And wild type would be excited by both of them, and of course VEX by only the violet one, and VEX by the blue one. And because they come up at different times, just slightly different times, microseconds, or maybe it's microseconds apart, on the impinging on the stream, the cells will be excited at slightly different times, and the electronics distinguishes very well the emission from one versus the other. And then, of course, the cells go into the droplets, and the droplets are charged and deflected in the standard way. But we can, with 2 different excitations, detect the cells or whatever labeled with one or the other, or both, for that matter. Okay, I'm now recording again. You've seen this slide already, and we're going to move on now to showing where GFP came from. Actual picture of a jellyfish. There it is. Beautiful jellyfish. They have a display of these in the Monterey Aquarium, and I also discovered they have them in the Boston Aquarium at this particular moment, because I was up there for a Gordon conference. Beautiful fish, beautiful jellyfish, and GFP is used so that these can luminesce. It transfers energy from GFP to whatever it is, luciferin, that, or luciferase, I guess, which puts out the actual light. Now, next slide. We're waiting. Stovall. Yep, I'm pretty sure that's Dick Stovall. It's an old picture of Dick Stovall with the FACS 1. And I should have put this in earlier, but I cannot disrupt the earlier part of the slideshow without recording everything from the beginning. And I don't think you want to hear that recorded again. I certainly don't want to do it again now. But there's Dick Stovall. Maybe it's Tim Merrill. That's what Lee thinks. Now we have, we're moving up to the very modern machine, the present, and it's going to be the future of FACS. So it's a 7-color, 8-parameter FACS analysis. And you see how the different laser lines are used for excitation, and the bandpass filters allow you to pick up the emissions, different kinds of emissions. I think the slide is self-explanatory, and you can study it all your desire. You're not going to move on. I'm just going to move on to the next slide. But you see we've got compensation on here, the very necessary compensations, leading to what we need to do now is to develop Flojo, which Mario Roederer had a lot to do with, Wayne Moore, of course, in the original algorithms, and the code was actually written by Treisler, Adam Treisler, and he set up the company called TreeStar, and you can actually buy it from the website, which he has for TreeStar. And an example of 8-color phenotyping of T cells. This is not a complete lecture about everything that we're doing, because it's covering, a lot of years, 30 years, and we're moving rather rapidly, and just picked out a few slides which I thought would be useful for you to see. So this is gating with a bunch of different reagents, and I think you can see how the gating works. I can't use a pointer to point it to you, but you have to follow the arrows, and you can see that first we took PBMC and picked out the CD3 cells. Sorry, we picked out the lymphocytes, and then we picked out the CD3, which is the upper second to the right box, and then gated that on CD4 and CD8 to the lower left of that box, and so on. You can see the gating's all done by the arrows. I think it's fairly self-explanatory. You see in the contour plots that we have, with some outlier plots included, looking therefore for dots as well as the actual contours. And the various reagents that we use are shown up there, and I haven't spelled out what they all stand for, but PE is phycoerythrin, TR, of course, is Texas red, APC is allophycocyanin, and Cy5, Cy7 are cyanine dyes, which are coupled with the PE or the APC, and it gives us a lot of different emissions and excitations. There it is. So you're probably all wondering, why do we need so many parameters, so many colors? Well, here's a partial example that to identify subpopulations requires at least 3 or 4 different, independent antigen measurements. Then looking at cytokines or galactosidase, beta-galactosidase, or glutathione, other things which we're going to come up with shortly in our slides, requires even more colors. And it's a great advantage to have multiple parameter sorters and analyzers, because fewer reagents are required. You can do it all in one tube, so less sample volume is needed. We can study pediatric patients for some of the applications that we've had. For example, looking at CV11B as a marker for ruling out sepsis, something which we're not going to show a slide on at this time, but maybe we'll talk about it in one of the sessions if we have a chance. The optical layout we have for 9-color 11-parameter machine, which is now in use, definitely in use in the FACS facility, and maybe some of you have or will see it in the FACS facility here at Stanford in the Beckman Center. And I'm not going to go through the actual block diagram, but it's quite informative if you just study it. So up to 93, there were stepwise advances in multi-parametric flow cytometry, FACS, in the Herzenberg lab, and they're spelled out here. We went from 4-color with 1 laser to 5-color with 2 laser. We also did 4-color with 2 laser at that particular time. 6-color with, I guess we did it much earlier in 84. We did 6-color with 2 laser, etc., and that allowed us to look at many of these different colors. So this was quite an important period during the early part of 93. But now, having a 10-color FACS or 10+ color FACS, there are analysis complications, things you have to do to make it all work. One is compensation is very important. There are different compensation matrices for each sample based on the combination of reagents used. You have to annotate. Database-like analysis is required for proper annotation. So you have to use databases for doing this, or construct databases. Gating. Gating is very important. So you may have 40 different gates for 10-color analysis of PBMC, or maybe of B cells in mouse work. A lot of this work with the PBMC, with AIDS samples or normals, controls, done by Mario Roederer, Steve DeRosa, probably leaving out a lot of people, Ewan, and earlier other people. Actually, I'm being a little bit tongue-tied here because I'm having to continue with the microphone here in front of me and the computer and controlling my own sound and all that. I hope it's coming out pretty well. Anyway, the gating hierarchy is what's fine. You can define 40 to over 100 different distinct cell types. And most of these cell types are subsets of another cell type. So generating a gating genealogy to represent this underlying biology turns out to be very important. And there are so many subsets, it requires a database-like approach to analyze these subsets, grouping, searching, retrieving analyses. And much of this is provided by FlowJo. And more of it's going to be provided as we continue doing software. Lee is leading a lot of this work, and we're working with a couple of other companies, including BD, cytometry, and MSA, a database company that we are newly forming a relationship with. Yeah. So I'm going to go back to some of the things that we can do ourselves, like how can FACS measure gene expression? Of course, immunofluorescence is one way, looking at the level of surface expression. It's simple, it's quantitative, requires antibodies. A lot of these antibodies, of course, are now available through Pharmagen BD, Pharmagen having been developed by Ernie Wong, and now sold to BD, and BD now has it as a part of its operation. So a lot of the antibodies do come still from the Pharmagen division of BD, Becton Dickinson. You can do biochemical analysis using fluorogenic substrates, like for beta-galactosidase, beta-glucuronidase, and different people have been involved in making, maybe I should particularly mention Gary Nolan, who was, and Steve Firing, who were involved in getting the beta-galactosidase assay online. I'll describe that in a bit with another slide. And things like FISH, fluorescence in situ hybridization, looking at any gene. It's quantitative. Of course, you can't do it with viable cells, and it's not particularly sensitive. Okay, the advantages of multi-parameter flow cytometry are many, and I've alluded to some of them previously, and I'm just going to leave this slide on. You can leave it on to read the whole thing at your leisure. Now, you know, Moore's law for increase in amount of capacity of memory and the cost going down, you know, twofold every, I don't know, 18 months, and going up twofold in terms of memory capacity every 18 months. That's the law of computers. Well, we have our own law here for FACS. It's Roederer's law. I think he named it Roederer's law. He made the slide, and it's kind of cute. And you can see how the number of dyes, number of colors which can be marked, which requires an increase in number of dyes, some of which he's been directly involved in helping to generate, and they come originally from Alan Wagner's efforts at Pittsburgh, and Biological Detection Systems, which was bought by Amersham, and now it's available through Amersham. And you can read the slide and see how, with different lasers, we've increased the number of colors just marvelously. And the number of colors doubles every 9 years, but I I think in the very recent years it's going up much more rapidly. We'll see. Next slide, but I can't remember them all. So why do we need so many colors? Well, information content increases geometrically with the number of colors. And I talked about some of this a bit in an earlier slide. Some information requires more colors. So if we want to distinguish naive and memory CD4 T cells, that needs more than 3 colors. And then we want to identify within these subsets, unique subsets, so which lineage they come from, CD4 versus CD8, what stages of differentiation they're at. And then we want to combine phenotypic and functional analyses, and that's all spelled out on the slide. And there should be a second part of it, but I didn't find the slide for this presentation. So, beta-galactosidase assay I promised you, FACS we call it, and it's an assay system for studies of somatic cell genetics and gene regulation. We're making a lot of use of FACS, of beta-galactosidase I should say. It's a very standard enzyme to use for measuring gene expression. And you can read through the descriptions on the slide. I'm going to change slides. Now, glutathione is another area in which we're very interested. I don't know why this slide looks so funny. The SH, it should be an SH of glutathione. It's a tripeptide. The level of glutathione goes down in AIDS in cells and in tissues. And we think that by restoring glutathione levels, you can help people a lot. And we find that you can restore glutathione with NAC and acetylcysteine. So much of our work in recent times has been concerned with that work. And it's a major interest of Lee's as well. And I think somebody may talk about it at the sessions for our anniversary here. Glutathione can be measured by using monochlorobimane, a non-fluorescent dye which goes into cells, into live cells, and there, under the action of glutathione S-transferase, it will be conjugated with MCB, so MCB will be conjugated with glutathione by GST to make a fluorescent adduct, which can be kept in cells by keeping the cells cool, and it excites in the UV and emits in the visible, and we call the compound glutathione S-bimane, GSB. By FACS, you can measure the glutathione in individual cells. You can measure different cell types by using surface antigen detection with the various CD antigens. So you see here that one cell, one type of cell, CD4 T cells, give you a median, and we plot the distribution. And now we look in various people who are HIV negative or HIV positive. You see quite a difference in the amount of glutathione in the blood, or in T cells, a CD4 type, or in monocytes. So CD8, CD4, T cells, and monocytes all have a nice correlation for the amount of glutathione measured by this GSP assay in the